Papanicolaou staining method. Pap staining method is a multichromatic staining method used in cytopathology, particularly for the screening and diagnosis of cervical cancer and other diseases. And this method can be performed either using progressive or regressive techniques. The pap staining progressive and regressive techniques, both methods use the same stains. Basically, hematoxylin, which is a nuclear stain, OG6, which is a cytoplasmic stain for keratin, EA, which is a mixture of eosin Y, light green, SF, and bismuth brown for cytoplasmic differentiation of cells. The regressive Papanicolaou staining technique. In regressive staining, cells are overstained and then differentiated to remove excess stain, allowing only the desired components to retain the color. And this technique can be performed as follows. Number 1, fixation. So the smears are fixed with 95% ethanol for 15 to 30 minutes. This is aimed at preserving cell morphology by coagulating proteins and preventing autolysis or drying artifacts. Second step, hydration through descending grades of alcohol to water. So the smears are placed in descending grades of alcohol, for example, 80% ethanol, 70% ethanol, then to water. The aim is to prepare the smear for aqueous staining. Step number 3, hematoxylin staining, commonly using Harris hematoxylin. Since it is regressive staining, the nuclei is overstained, ensuring that all nuclear material is intensely stained. Then after, the smears are rinsed in water to remove the excess hematoxylin. Next step, differentiation using 1% acid alcohol for 2 to 3 seconds. The intent is to remove the excess hematoxylin from background cytoplasm while leaving the nuclei stained, which we call controlled destaining. The next step, bluing, using any bluing agent like running tap water. We can use lithium carbonate or Scott's tap water. The aim is to change the reddish hematoxylin to a crisp blue-purple color, improving contrast and visibility. Next, dehydration through grades of alcohol, for example, 80% and 90%. So these ascending grades of alcohol prepare the cells for alcohol-soluble counterstains, specifically OG6 and EA. Next, we stain with OG6 or orange G6 for 2 minutes to stain the keratin-rich cells, for example, superficial squamous cells, orange. Rinse in 2 changes of 95% ethanol. The major reason is number 1, remove excess OG6 and prevent carryover to EA, and also it helps maintain alcohol environment for the next stain since it is alcohol based. Next step, we stain with EA, and there are many variants of eosin azure. For example, EA36, we have EA50. We stain for 3 minutes. The reason is to differentiate the cell types. So EA50, as we said earlier, has eosin Y, which stains the mature squamous cells pink, and light green SF, which stains metabolically active cells, for example, parabasal cells and intermediate cells blue-green. dehydration through absolute alcohol. The smears are passed through 95% ethanol for 2 minutes and 100% ethanol for 1 minute. The reason is to remove water from the smears, since water is immiscible with subsequent agents that are going to be used. For example, clearing agents like xylene and the mounting media, but also prepare the smears for clearing. Next step is clearing in xylene or any clearing agent. And the major reason is to make the smear transparent for microscopic examination. Finally, mounting with any mounting media that is miscible with a clearing agent. For example, once we have used the clearing agent like xylene, the mounting media that is miscible with the xylene is DPX. And the major reason is to preserve the stained smear and provide a permanent preparation. Outcome of a good pap stain. 1. The nuclei should appear crispy blue to purple with clear chromatin patterns. Number 2. The cytoplasm of superficial squamous cells will appear pink to orange. And number 3. The intermediate or the parabasal cells will appear with a blue or green cytoplasm due to the effect of EA. In evaluating the staining quality of a pap smear, the following parameters are considered. Number 1, nuclear chromatin visibility. So after staining in hematoxylin, the chromatin dots within the nucleus should be clearly visible under a microscope, and in case of active mitosis, it should be clearly seen. Number 2, cytoplasmic differential counterstaining. So when counterstained, the cytoplasm of the cell takes up different colors of the stain used. For example, OG6 stains the cytoplasm of old and keratinized cells orange, and EA will stain the cytoplasm of young cells blue or green respectively. Number 3, cytoplasmic transparency. The cytoplasm of the overlapping cells should be transparent such that the cytoplasm of each cell is read independently.